Greetings all. I am Dr. Sathya, Assistant Professor, Department of Botany, Bishop Heber College. Today we are going to discuss about the ultrastructure of plant cell. The main difference between the plant cell and the animal cell is cell wall. Okay, so today so many interesting things we are going to learn about the plant cell. So here the plant cell wall structure. Do you imagine if we don't have the plant cell or any cell doesn't have cell wall? What happened? We doesn't have the shape. So that the same thing, the plant cell wall, which have the typical component of cell wall that fixes the size, shape of the mature plant cell. The kind of cell wall present determines the texture of the tissue. If it is the thin wall, the texture is very soft nature. If it is the thick wall, the cell which is very rough in nature. So based on the cell wall, it determines the type of the cell and the consistency of the cell. And what is the main function of cell wall? The cell wall, the main function, that is the mechanical function and the metabolic activity and nourishment and absorption or secretion. So what is the mechanical function? Like the skeleton around each cell. The cell wall gives the skeleton, it surrounds the cell, so it gives the wonderful shape to the cell, and it determines the shape and size of the cell. Based on the cell wall, it determines the minimal size or the maximal size, and determines the limits of expansion of water uptake. So if it is the permeability of the cell wall, it determines the uptake of water. And metabolic activity, the cell wall, which is living part of the cell, which receives the signals such as the hormones. If it secretes the hormone from one cell to another cell, it receives the signal from the neighboring cell and it communicates to secreting and do the function. And wall signals transmitted through the plasma lemma, that is the plasma membrane, and into the cytoplasm, and it gives the biochemical responses to the cell. And in persimmon, you know that it is the wonderful fruit. In that, the cell wall only gives the nourishment to the fruit. And absorption and secretion in root hairs and rhizoids, which have the absorption and secretion function. And the cell wall very much in thickness and based on the age of the cell. So if it is the young cell, the wall is very thin. If it is the matured cell, the wall is very thick. The cell wall is the complex structure, but it is very interesting structure. See, into the ultrastructure, we have the primary cell wall, intercellular substance, and secondary cell wall. What is the primary cell wall? It consists of only one layer. Coming to the intercellular substance, so it is very important thing, that is the middle lamella. Middle lamella is nothing but it is giving the cement-like bonding to the neighboring cells. And the secondary cell wall, which is made up of many layers, it frequently most of the cell wall which have the 3 layers. What are the 3 layers? We are going to see. Okay, see, this is the ultrastructure of cell wall. See, this is the primary wall, this is the secondary wall, and this is the plasma membrane. Okay, so we know that middle lamella, in between 2 cells, it is like the cement wall. So see here the yellow color layer which gives the cement-like material which act as the cement, that is to connect the 2 adjacent cells. So here it is the plasma primary wall, that is the outer wall, and the secondary wall inner to the primary wall, and the innermost wall is plasma membrane. And what is the primary cell wall? The structure of primary cell wall, it is very interesting and the complex thing. So what is the primary cell wall? The 3 major polysaccharides which is located in primary cell wall. So the cellulose is the main component. We learn cell wall is made up of cellulose. How the cellulose is made of, compose the cell wall, the primary cell wall. So which bonds the beta 1,4 linkage and then long unbranched linear chain of glucose with crystalline properties because of arrangements of cellulose. So based on the arrangement of cellulose which forms the primary cell wall. So the important thing is the cellulose which composed of microfibrils, macrofibrils. This microfibrils is made up of the micelles, which is the smaller unit of cell wall. So the smaller unit is micelles, which micelles are composed to make the microfibrils and microfibrils to compose to make the macrofibrils. See, this is the clear image of cell wall. So here it is the micelles, that is the chain of cellulose molecules. Thus molecules composed to form the microfibrils. This microfibrils to form the bunch of microfibrils to compose to form the macrofibrils. This macrofibrils forms the cellulose fiber. Now you clearly understand about the cellulose, how to cellulose arranged in the cell wall? Yes? Yes. The microfibrils, the macrofibrils and the cellulose. So very interesting component. This is the structure of micelles, that is the hydrophobic chain and hydrophilic head, that is the water loving head and water hating chain. So this is the structure of micelles. So here the same thing, the bonds which is in between the micelles and H bonds and crystalline arrangements. This micelles forms the fibrils and it forms the amorphous matrix and then it forms the cellulose. Okay. And the second component is hemicellulose. The hemicellulose, that is highly branched long chains. So here it is compared to the cellulose that is non-branched, here it is the highly branched glucose, that is xyloglucans and xylans. So xyloglucans, the microfibrils coated with the fibrous hemicellulose. Here it is the fibrous hemicellulose and it is the xyloglucan, made up of xyloglucan, and in between that which have the hydrogen bonds with the cellulose, and xyloglucans is turned chemically bonded to another hemicellulose. So this serves as the cross link between the pectin molecules. Now you have to clear image. So imagine this is the clear image of primary cell wall. So this is the hemicellulose. In between this hemicellulose which have the structural proteins and pectins, and this is the cellulose microfibrils. So is it clear? Yes. Okay. And next third component is pectin. The first one is cellulose and then second one is hemicellulose and the third component is pectin. And then cellulose and hemicellulose embedded in that and form a separate network that integrates with the cellulose hemicellulose networks. Previous image explains how the pectin integrated with the cellulose and hemicellulose. Okay. And it is middle lamella, in between that which is middle lamella region between the cells and composed of pectin and glue cells together. So in between the hydrophilic holds up 65% of water in primary walls. So what is the function of pectin? Which gives cell adhesion and regulate the porosity, that is the small pores. It regulates the small pores which have to used for the transport of the cell materials. So I am coming to the proteins. What are the proteins? Are peroxidases, cellulases, pectinases, phosphatases. So many proteins are located in the primary cell wall, and hydroxyproline and rare amino acid found in the plasma membrane which is bound onto the polysaccharides, and it is forming the glycoproteins, that is the rich protein located in the cell wall. So do you don't forget that word. Glycoproteins are rich proteins which is located in the cell wall, and the function is fall expansion and involved in the growth and development. So again I say that the middle lamella and primary cell wall and plasma membrane, and see the soluble proteins are here. So in between the hemicellulose and cellulose, the pectins are wire-like structure, it is intertransmitted, and here it is the proteins, soluble proteins are located. And then lipids, cutin and subrins and waxes. These are the types of lipids which is present in the cell wall and found in cuticle. Cutin, we know that the suberin is waterproofing cork cells which is present in peridot and also found in Casparian strips. Do you know the anatomy of dicot stem and root? The root cells, the protoxylem part which have to passes the water molecules easily absorbed. That cells are passage cells, and inter between the two protoxylem the cell wall is very thick in nature, that is the Casparian strips. So in Casparian strips the suberin is present. Because of thickness the suberin is located there, and waxes also coated on the plasma cell wall, primary cell wall. And the cell wall layers, and here it is the protoplasm form its wall in the outside to inward, and so the oldest layer wall is outermost position and the innermost is young one. So here it is the secondary layers, secondary structure of cell wall. So now we complete the primary wall, and now it is the secondary wall. In secondary structure is very beautiful arrangements of microfibril cellulose. So how it is arranged? S1, S2, S3. The three layers, important layers, based on the arrangements of cellulose which formed on the S1, S2 and S3. What are the arrangements? So coming to the secondary cell wall. The secondary cell wall which have the S1, S2, S3. So what is S1? That is horizontal orientation of microfibrils. Now we know that microfibrils, which is made up of micelles. So these group of microfibrils forms the macro and two cellulose, isn't it? So here it is the horizontal orientation of microfibrils. And the S2 thickest and the S3 is very thin and lacking, sometimes called a tertiary wall. But it is horizontal. S2 is opposite to the S1, it is vertical in nature. The arrangements of microfibrils are vertical. So the different orientations of the cellulose and microfibrils in the three layers. So based on the different orientation of the microfibrils which decides the different layers of secondary cell wall. Okay. So here it is the orientation of microfibrils. This is the S1 layer. S1 is horizontally arranged microfibrils. This is the S2 layer, that is vertically arranged microfibrils, and S3 layer horizontally at larger angle than S1. This is the difference between the three layers. Based on the arrangements, the secondary cell wall is composed. And this is the cross section, how it is the primary S1, S2, S3 layers are arranged here. And coming to the image, this is the primary cell wall, and cellulose is coated by the hemicellulose, and this is the lignin layer. So it clearly shows that the arrangements of different layers of cell wall. So intercellular spaces, that is nature which is occurs in the mature tissues and may be found in the meristematic tissue intensive respiration. So may function as the containers for secreted materials. So that based on the intercellular space is classified into schizogenous and lysogenous. What is schizogenous? That is the most common type. So based on the enzymes which have to remove the pectin and which forms the spaces from the corner to the spreads to the other parts of the cell. That is the schizogenous. And what is lysogenous cells? The lysogenous results from the breakdown of entire cells and forms the space. That is the two different types of spaces, intercellular spaces. So intercellular spaces based on the arrangements of removal of pectins, that is cystogenous, and the entire cells, the results breakdown of entire cells, that is lysogenous. Do you understand clear? Yes. And in that important thing is pits. The depression occurs in the secondary wall, that is called pits. The pits are classified into two types. One is simple pits, another one is bordered pits. So pit cavity remains the same diameter, that is called the simple pits, and which have the aperture like secondary cell wall, which have arches and pit chamber. This is the pit chamber, the outer aperture, and this is the inner aperture. So which forms the entire pit aperture. So which forms the border and inner aperture. So this is bordered pit and simple pit. So pit pass. So how the secondary wall is located here? This is the pit cavity. This gives the simple pit, and this is the secondary bordered pits. Okay. So do you understand the difference between the simple pit and the bordered pits? Yes. And the next component is middle lamella. So it is the thin, amorphous and cementing layer between the two adjacent cells. As I said, this is the cementing layer in between the two adjacent cells. So middle lamella is the first layer which is deposited at the time of cytokinins, and it is just like brickwork of the common wall between two adjacent rooms. So middle lamella is absent on the outer side of surface cells, and it is made up of calcium and magnesium pectins. So what is the main function? It is act as the cement layer of adjacent cells. And see the canus, this is the primary wall, and the secondary wall, this is the cytoplasm and the plasma membrane. So each gaps which is surrounded by the plasma desmata. So that here the word is plasma desmata, that is basically holes in the middle lamella and primary wall lined with the plasma membrane. And plasma desmata, as I said, it forms the fusion of vesicle into the cell plate. So here it is the plasma desmata, the line in between the two adjacent cells, the green which shows the two cells. This is the nucleus and this is the adjacent cell nucleus, and here it is the plasma desmata. So it acts as the cement layer which connects the in between of two adjacent cells. Thank you.